Warm welcome to all to our channel World of Zoology. In the biotechnology series, we'll start next point regarding achievement of biotechnology in the medicines and industry. We'll consider first achievement in the medicine, that is in the medical field. Welfare of mankind through improvement of human health has been one of the most significant application of biotechnology in the medical field. Development of newer drugs, therapeutic agents in the form of enzymes and other proteins, as well as the gene therapy and stem cell therapy, have been recorded as a breakthrough strategies in combating some of the diseases that had been almost incurable in the past. One of the major techniques has been developed, the recombinant DNA technology, that involves constructing DNA molecule, bringing together genetic material from more than one source, which then possess a sequence that otherwise could not be found in the given organism. The technique has been effective in various ways, one of which being production of pharmaceutical significant compounds using recombinant organisms. These are some of the achievements of the biotechnology, where biological products are produced by using microorganism or animal sources or the living cells. Pharmaceutical preparations are prepared by using biotechnological techniques. Then for diagnostic purpose also, biotechnological techniques are used, where biomarkers are also one of them. Then cosmetics and perfumes are produced by using biotechnological techniques. In the lab also, laboratories we also use biotechnological techniques. Then here the medical devices such as pacemakers, which are working on the biotechnology, medicinals and botanical products that are playing important role by using biotechnological techniques. And thus, the achievement of biotechnological technique is shown in this slide. Biomarkers for disease detection. Some of the proteins are specifically expressed by the cells of tissues under certain disease conditions. Hence, these proteins are called markers, or more appropriately biomarkers, which are the indicators of that particular disease and hence have potential as a diagnostic tool. That means if the particular protein is present, the particular disease is used to diagnose. Many biotechnological industries are involved in the research, that is R&D department of that particular industry, pertaining to the development of techniques that can help to find such biomarkers for early detection of various diseases so that the disease control is possible. This is a growing trend of research in finding potential biomarkers for tuberculosis, that what we call as TB, AIDS and other infectious diseases. Various genetically inherited diseases can be diagnosed through a gene sequence analyzing by using restriction fragment length polymerism. This has helped to understand the gene mutation that leads to various genetic disorders and afflictions like sickle cell anemia, cystic fibrosis, hemophilia, etc. That is the use for understanding the gene mutations such as cystic fibrosis, sickle cell anemia, that is particular shape of the RBC which is like a sickle, and the disease is sickle cell anemia. Then hemophilia, that is low level of the hemoglobin, or hemoglobin content is less below the normal level. This has facilitated developing therapeutic proteins or their transgenes that help in the treatment of these to a greater extent. Then the next point, that is gene therapy. All therapy means curative treatment here. Amongst the various strategies involved in treating the genetic diseases, gene therapy has emerged as a consequence of recombinant DNA technology. With the help of this genetic engineering technique, a larger number of enzymes and structural proteins can now be made available for therapeutic use in diseases like severe combined immunodeficiency disorder, that is SCID, and cystic fibrosis, then insulin-dependent diabetes, etc. Here, the diagram is shown where the production of the insulin by using microorganism with the help of the recombinant DNA technology is shown, and a particular plasmid molecule is from the bacteria is modified by using recombinant technology. A desired gene is inserted into a plasmid. First of all, you must remember the plasmid. Plasmid means an extra chromosomal part of that particular organism having capacity to multiplicate and introduce that particular gene into the particular bacterial genetic material. Plasmid is a small extra chromosomal DNA molecule within the cell that is physically separated from the chromosomal DNA and can replicate independently. There are most commonly found material as a small circular double-stranded DNA molecules in case of the bacteria, and these plasmids are also present in some of the eukaryotic cells sometimes. Here, these plasmids are reconstructed by the desired gene, and the particular part of the gene is isolated and introduced by cutting the particular segment with the help of the restriction enzyme, and then the patchwork-like function is carried out by using recombinant DNA technology, and the desired gene is inserted back into the microorganism where it gets incorporated into the genetic material of the particular bacterium and multiply and produce the copies of the human desired gene. Here human insulin is prepared by using microorganism, and then it is extracted, purified, and used as a human injection for injecting purpose, which is shown in this slide. Vaccines and therapeutic antibodies. Some of these vaccines synthesized through the genetic engineering are recombinant vaccine for hepatitis B virus, rabies virus, foot and mouth disease virus in case of the cattle. The medical biotechnology has tremendous scope in the pharmaceutical industries in the production of the antibiotics, which are potential antimicrobial agents against some of the disease-resistant strains of the pathogens. A recombinant strain of the bacteria Streptococcus mutans has been developed and is under clinical trial for its efficiency in reducing tooth decay. Then the next point, that is stem cell technology. Human embryonic cells, that is ES cell, human embryonic stem cells, that is ES cells from the blastocyst stage, which are undifferentiated cells, have been isolated and successfully cultured. Here remember that cells in the blastocyst cells are undifferentiated, and therefore they can develop any type of cells in our body which are required. Owing to this pluripotent nature, they can differentiate into any other cell type. Means the ES cells are pluripotent in nature, and these are separated cells of the blastocyst or blastocyst stage. Thus, through the transgenesis, this can be manipulated in order to differentiate into particular cell type, which can then be introduced into host tissue to replace the particular defective cells to carry out the function in normal fashion. Thus, this technique has the potential to cure some of the genetic disorders like sickle cell anemia, in which the stem cells from the umbilical cord of the blood could be used. Here, umbilical cord means what we term it as nal in Marathi, where there is a connection in between the mother and fetus that carries the blood, and that umbilical cord blood is the source of the ES cell, that is undifferentiated cell. Now, the next point is the Human Genome Project, that is HGC project, HGP we can say. Human Genome Project was initiated in 1990 by US Department of Energy, which was the international collaborative efforts with 15-year plan to identify, sequence and map all human genes. Analysis of the human genome has thus provided a backbone for further research in biotechnology, where recombinant DNA and transgenesis techniques have been standardized for various genes that have been successfully manipulated for human welfare. Understanding the gene sequence has given better insight into various mutants or mutations in case of the genetic diseases. The same has also led to breakthrough in discoveries of the drug targeting the specific mutant genes causing cancer. The next field is the industry. The application of biotechnology in the industry can be majorly attributed to the use of the microorganisms that have helped to create the enzymes, antibiotics, simplify manufacturing and production processes of many food products, including the dairy industry, then leather industry, whatnot. We can say the various types of techniques which are used in each and every industry so as to improve the quality of the product. Have also found scope in environmental conservation through bioremediation. One example here can be explained, that is use of the particular strain of the microorganism so as to digest the oil which is sprayed by the oil spillage from the in the sea, and so these microorganisms can digest the oil that forms a cover over the surface of the ocean, and thus these microorganisms are used for bioremediation. Fermentation, that is one of the first point. Microbes are used for making bread, yogurt, cheese, as well as beverages such as beer, wine, champagne and liquors. Due to the anaerobic metabolism that is carried out by various microorganisms, the byproduct form is either lactic acid or ethanol. This process is called as lactic acid fermentation or alcohol fermentation, depending upon the end product which is formed. Alcohol fermentation has been widely used in beverages, where traditionally the yeast strain Saccharomyces cerevisiae is widely used. Here again, depending upon the bottom fermentation or the top fermentation, the particular type of beer which is produced, it varies and the quality is accordingly. Various food products like bread, idli are also produced by the fermented dough or batter in which the yeast is used. Hence, it is called as baker's yeast. Foods. Traditionally, to make cheese, even the curd, milk is coagulated by adding curd or lactic acid, that is lemon juice. Lactic acid produced by the fermentation leads to coagulation of the milk by curding. Also finds its application in processing of the cheese, then sour milk, sour cream, yogurt, etc. In dairy industry, the best tasting cheese is produced by adding enzyme renin, which is commercially produced by recombinant DNA technology, where this protein is expressed in bacterial or fungal cells. Recombinant renin so produced is called chymosin, is the first recombinant DNA food ingredient to be approved by the FDA USA. Biotechnology in dairy industry, which is shown in this diagram, where the various milk products are shown, which are used to prepare the cheese and the milk products. Pharmaceuticals, that is the next field. Recombinant DNA technology has helped in the big way in the manufacture of various therapeutic proteins such as insulin, which is also one of the protein which is used to treat type 1 diabetes. that is insulin-dependent diabetes mellitus. Production of the vaccine using recombinant DNA has been successful in case of the hepatitis B. In 2005, pharmaceuticals company Merck received FDA approval of the Grandcell, a recombinant subunit vaccine against cervical cancer, which is the first cancer vaccine to be approved by FDA. This vaccine targets 4 specific strains of the human papilloma virus, that is HPV, which is causative agent for approximately 70% of cervical cancer in women. Here the vaccine which is produced on that, and also the vaccine, the efforts which are being in progress for the production of the vaccine on COVID-19. Thank you for watching this video. If you like the video, do like, share for the benefit of the maximum students, and subscribe the channel World of Zoology. Thank you once again.